Hey guys, so I wanted to go over real quickly with you the why of our next step in our workflow. So remember in biotech, what I've been talking about is having really consistent and longer workflows where the input in one lab will become an output or a desired product. We'll then take that desired product that we get at the end of one lab, and that will be the input into a lab downstream or the next lab, right? So an example of this is you guys just completed an input where the input was your cheek cells, right? And we had a desired molecule within those cheek cells. That input was our cheek cells. We processed it through a process called cell lysis, which just means we broke open the cells and we isolated the DNA. So what our output of that last lab was 150 microliters of genomic DNA, just meaning all of your DNA, all of your genes, right? So that leads to a problem, as you just read in the lab that you're doing for next week, in glowing confidence, that lab, right? The problem is we need some sort of way of knowing whether or not the thing that we wanted to get, in this case DNA, is actually in that invisible solution. So that's the problem that we're working with, right? DNA is invisible in solution. We could see it before coming out of things like strawberries or cheek cells because we precipitated it. But we didn't do that step this last time, right? We just left it in solution. And those negative charges on phosphates, that's part of the backbone of DNA, causes it to disappear in this polar solution, in this polar liquid called water, right? So we can't see it. So we have to visualize it somehow. It would really stink if we went through this long workflow all term long, and what we found out was like, well, we just never had any DNA in the first place, and we screwed up our very first step. So we want to confirm that, and we want to confirm something like this. We call it an assay. So this week's lab is going to focus just on visualizing DNA because it's invisible in solution. There's a couple of really common chemicals in the lab that will allow you to visualize this invisible bit of DNA. One is called ethidium bromide, which is here. The other is called CyberSafe, and there's other types as well. But these are the two that we primarily work with in the lab here, and labs across the world really use these couple. CyberSafe will give you sort of like a green color. What we're going to use is something called ethidium bromide. And I want to make sure that I point this out again. It was in your lab and in the reading that you just did. But this stuff is carcinogenic. It's really, really nasty. It cannot go down the sink. You must wear gloves and eye protection next week. You're working with really nasty stuff, right? Some stuff that we can do with ethidium bromide is we can actually add it to something called a gel, which I have an image of right here. This is something that you guys will be doing on later on in the course. When we want to separate DNA based on its size, that will be something that's really great and important for us. Really what this stuff does ultimately, though, is this is what the ethidium bromide actually looks like, the chemical. And after this week's lessons, you guys should understand that this is the general structure of DNA, right? It's got this sugar phosphate backbone, and then it's also got these nitrogenous bases sticking towards the middle and joined up like this, right? What ethidium bromide is really great at doing is it will do something called intercalating. It will actually fit, which is what this is showing you, right? Like in this double helix, you're seeing this ethidium bromide actually fitting in between the bases. And as that chemical fits in between the bases, it's going to interact with light differently than what it normally would. And what that allows us to do is visualize this DNA. So what you're seeing over here is DNA that's separated by how big it is. It's actually an experiment. This is a gel that I ran here in the lab at Castleview. And we'll talk about, what all this information means. Right now it doesn't matter. All I want you to see is what happens to DNA when we add ethidium bromide to it. It goes from invisible to this pink-orange color. So all these, you guys see these really fluorescent lines that you're seeing right here on this image? That's DNA. So what we're going to attempt to do in lab this week is visualize DNA by staining it with ethidium bromide. And then what that will allow us to do is confirm whether or not you actually got DNA isolated in your invisible solution that was your output from our last lab. So hopefully that makes sense, guys. Go ahead and read through that thing. By the way, I have this piece of equipment right next to me. I almost forgot to show you. We're going to use something called a transilluminator in order to see our DNA. This is going to shoot some UV light at it. This is what it looks like, right? Some powerful UV light will interact with that ethidium bromide that's fit or intercalated within the DNA. It should glow. If we've got glowing, we know you have DNA. We can continue along downstream. If you don't have DNA, well then, whoops, we got to go back and do some error analysis with the last lab that you did. And that's science. Okay, guys, I'll see you in the lab first thing next week. So my A group, you're in the lab Monday. B group, you're in the lab Tuesday.